Hello, everyone. My name is Dr. Qian Wu. I'm a group leader from University of Leeds, working on the structural mechanism of DNA repair pathways. I'm also one of the panel members of the research area of signaling of Biochemical Society. Today, on behalf of the Biochemical Society and Portland Press, to welcome you all to this webinar, which is part of our Biochemistry Focus webinar series. Topic in this series including different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions directly through the text, and we welcome suggestions for future topic and speaker to feature in our webinar series. And you can find out more information in our website. In today's webinar, you will hear talk from Professor Dan Rigden about his perspective for AlphaFold 2 that enable protein structure prediction with unprecedented accuracy. So during the webinar, you can send in your questions during the talk by typing it into the question box as shown on the screen. And there will be a break in the middle of the talk to answering some of the questions. And we will also to answer as many questions in the end when the presentation finish. So now let me properly introduce today's speaker. Professor Rigden did his undergrad study in the University of Oxford, followed with PhD in University of Edinburgh. After postdoc training from 2004, he established his group in the University of Liverpool and now a professor of protein bioinformatics at the Institute of System, Molecular and Integrative Biology. Professor Rigden has a 30 years experience in bioinformatics approach to protein structural function evolution, and this covers aspect protein function annotations and interpretation by domain, motif, and structure-based approaches. He was an early adopter of the ab initio protein structural modeling approaches for protein function prediction and molecular replacement. And he is one of the assessors at the CASP14 in 2020, looking at the performance of cutting-edge protein structure prediction methods, including AlphaFold 2, and he is continuing his role of assessor role in the CASP15 this year. So now it's my great pleasure to welcome Professor Rigden for his webinar.
Share my screen. How's that looking?
Yeah, we can see you.
Okay, great. Well, very nice to be here. I can't see any of you, of course, but I'm just going to imagine hundreds of happy, engaged faces. So I'll be telling you about AlphaFold. Just let me organize my windows here. So that's my title, AlphaFold Modeling, Access, Uses and Limitations. So this is the outline. I'm going to give you a very brief history of how we got to AlphaFold 2. I think that's good to set it in a bit of context. Then we'll talk about how good the models are that it makes. I will talk a little bit about some other aspects, some other data that AlphaFold produces alongside the coordinates, which is very important to help you interpret the model, decide how trustworthy it is. Then we'll talk about ways to get AlphaFold models of proteins that you're interested in. There's now a database, you'll probably be aware, we'll talk about that, but you may need to make your own models, and that can be as easy as using a web page, or you might want to do it locally. And at this point, I'll specifically talk about some limitations of AlphaFold models and specific limitations about the database, because although you'll see it's an absolutely fantastic resource and an amazing program, you have to bear in mind the limitations as well. At that point, we'll break for a few questions just so you can recharge your batteries. And then we'll go on to talk about what AlphaFold models can, maybe can't be used for, and the kind of ecosystem that's grown up around AlphaFold, some of the ways in which people have used it, even for purposes it wasn't really designed for, and some of the competitors, in a sense, that have come along to plug some of the gaps where AlphaFold couldn't do particular tasks. And so that will be taking in particularly structural biology, where it's a very key part of the landscape for structural biologists these days. But we'll also talk about AlphaFold for predicting interactions with small molecules or with other proteins, and what that tells us about protein function. So to start right at the very beginning, we often have protein sequence. We would like a model of it, and historically we can do template-based or homology modeling. This involves matching our sequence to a known structure in the PDB that we believe to have the same overall fold as our target. And then it's a relatively straightforward, well-established, pretty reliable way of generating a model. Where, of course, you don't have a convenient structure in the PDB to use as a template, then you're in ab initio or de novo modeling territory. And this is a totally different area that historically has been much more problematic and much slower developing. So taking as a starting point 1994, this was when the first CASP exercise was. You heard a little bit about CASP in the introduction. CASP is critical assessment of structure prediction. So this is a group of people who every 2 years invite predicting groups to make blind models of new structures that are about to come out. And then these are structures that are not yet present in the PDB, and so we can see for real how well the models work. For ab initio modeling, in the very beginning, they didn't work at all. And that's a quote from CAS1 in 1994. At that stage, people were using versions of molecular dynamics on very small proteins to try and fold them up, and it basically didn't work at all. At the beginning of this century, the first method that really did work at all, called fragment assembly, was introduced, pioneered by David Baker and the Rosetta software. And here we basically take a sequence and we find little chunks that we believe to be possibly representative of the local structure, and we stitch them together in a simulation, something like this. And this basically worked reasonably well, but for very small proteins. Of course, the reason we're all here was 2020, CAS14, when this is a holy grail, when AlphaFold 2 produced some models of really unprecedented quality. And this is a quote from the organizers, that in some cases, the structure predictions were really at the accuracy you would expect of an experimental structure. So there are differences between crystal structures of the same protein. Those differences were of the kind of magnitude of the difference between the AlphaFold prediction and a crystal structure. So essentially, they're as good as the crystal structure in some cases. And this is a very important qualification that we'll come back to throughout. So the development of ab initio modeling is really a story told through different CASPs from 1994 to the one where AlphaFold 2 broke onto the scene. This is a very nice graph from the CASP organizer, which shows the quality of the models that could be produced here, ranging from easy models, that consider those as basically homology modeling, to difficult modeling cases, which are basically ab initio modeling. And you can see right from the beginning, we could make reasonably good homology models, but in the very beginning, we couldn't make ab initio models at all. Progress was very slow. Actually, at CASP 13, there was already something of a breakthrough. That was AlphaFold 1. But at CASP 14, AlphaFold 2 has produced these models shown by these large circles of very high quality models across the range of easy targets right up to difficult targets. So almost as good performance with very hard cases as with the easy cases before, and well ahead of the next best method, as you can see. And just a note in passing, I quite like this perspective from the boss of the EBI on where we got to, or how AlphaFold really came on the scene, what was necessary. Of course, what was necessary was the deep mind scientists, very talented people with access to very powerful machines, but also not to forget that they trained those powerful machines on the PDB. So it was actually really important, essential, that the PDB from the beginning was an open data resource sharing the structural information. And it's only when it got to, it's only with the benefit of a large quantity of information in the PDB from decades of work by crystallographers and other structural biologists, the data are there to train the network on and produce AlphaFold 2. And then thirdly, just understanding the importance of CASP as a way to sort of monitor progress, incentivize groups, spot areas that need improvement year on year, and really monitor and push groups to do ever better performance. So these three things really come together, I think. So what does AlphaFold 2 look like? This is a representation of a complicated deep learning network, which is what it is. You take your, I won't go through the query details really. I'll just point out the different kinds of information that are derived from the sequence. It produces an alignment, a multiple sequence alignment. It produces covariance. It uses templates. That's one of the points I want to make. So in a sense, AlphaFold 2 is kind of merging the homology modeling and the ab initio modeling scene, where templates are available. They feed into the calculation, but where they're not, it can make very good models nevertheless. It also works by extracting covariance information. I'll take a moment just to explain that, because this comes back later in the talk. So covariance is a way of analyzing a multiple sequence alignment, which allows you to extract predictions of which residues within the alignment, purely based on sequence information, are actually close together in 3D space. And actually you can go beyond that with advanced methods and predict how likely two residues are to be. 8 angstroms apart, 10 angstroms apart, and so on. That information can be extracted from this multiple sequence alignment, provided that multiple sequence alignment is sufficiently large number of sequences, and also diverse. So it's no use having an alignment that's 23 very similar sequences. You need a diversity of sequences. So this will come back a bit later on. So it's kind of important that this multiple sequence alignment allows AlphaFold 2 to extract covariance information, and that's actually important in some cases for good quality modeling. The only other thing I wanted to pick out here is that there's an iterative element to the AlphaFold 2 process. It makes some initial models and then feeds that information from them back in. And by default, it does this so-called recycling 3 times, but that is something you can change. And in harder cases, it can be advantageous to try more recycling, push it a bit harder to get a better model. So AlphaFold 2 was published July 21. It's fair to say a lot of people, including myself, were a bit skeptical about whether it was going to be published quickly, whether the code was going to be made open source. Both of those happened very quickly, so absolute kudos to DeepMind for making it available so quickly. Here are some more examples of the amazing high quality models that they could produce with AlphaFold. So very large things to an RMSD of 2.2, pretty good. Smaller things to a very low RMSD, very high quality. This example here just shows that even though AlphaFold 2 doesn't know about ligands, so this is a metal binding site, AlphaFold 2 doesn't know about metals, that metal binding site was modeled well, and we'll come back to ligands later on. This is perhaps the most important figure. It shows 2 things. What we've got here is the actual quality on the y-axis, and we've got some predicted quality measurements that come out of AlphaFold on the x-axis. That's this PLDDT. 2 things here. If you look at these points, which each of one represents a model that was made and compared to a crystal structure to see how good the model was, you'll see that these points cluster very high up on this range, which means that on average, typically, we're getting very high quality models. But you'll also see that this blue line here, which is a linear fit against the actual quality versus the predicted quality, it follows this one-to-one line very, very closely. And what that means is that the predicted quality that AlphaFold gives you, this PLDDT score, it gives you that on a per residue level. That is a very, very good predictor of the actual quality. So not only is AlphaFold 2 giving you very high quality models, most typically, it's also correctly identifying regions that are high quality and also correctly identifying regions that are low quality. And that's equally important, as you'll see. So yeah, AlphaFold 2 produces a coordinate, it produces these local measurements of quality. We'll come on to this third one in just a second. So a little bit more about the predicted PLDDT. So there are some color schemes that have become kind of default, and you'll see these over and over again when you enter the AlphaFold world. So this is an AlphaFold 2 model from the database that we'll talk about a bit later on. And this is the kind of color scheme that's used there. So regions, residues that have a high, very high predicted quality are dark blue. Then we've got light blue, yellow, and orange. And so you look at this model here, you say, okay, this folded domain here is very high quality or actually confident, but these two tails here, the N and the C termini, those are predicted to be low quality. Got another model here, we've got 6 beautiful dark blue domains, but then at one end we've got what I've taken to call red spaghetti, which is this kind of very low quality scrambled ball of string kind of style structure, which is not reliably predicted, of course. You know, no protein actually looks like that. So if you see a region that's low confidence from AlphaFold, what does that mean? Well, sometimes it simply means that the model is bad. And here's a paper where they found some bad predictions from AlphaFold. In this case, the purple structure is the known protein, and the rest is what AlphaFold produce. And you can see it's in no way correct. But at least you will notice that it's recognized that this is low quality prediction. So the orange and the yellow coloring here is saying this is pretty low quality model, and indeed that's correct. So when it fails, it kind of knows that it's failed and tells you it's failed, and that's very important. And here's another protein that should be folded up, but for some reason AlphaFold couldn't model it. Interestingly, when you look across different species and you say, what proportion of the protein in the proteome of human, these other organisms here, is high quality prediction, we get quite noticeable differences. So for bacteria, we get very high quality predictions. For human, we get much more low quality predictions. And there's an interesting reason for that, which is that these low confidence regions are not simply where the modeling has gone wrong, but they're actually where the modeling, in a sense, has, where there isn't a single correct structure to be modeled. Let me try and explain that a bit better. You probably know that portions of proteins, even whole proteins, can be what's called intrinsically disordered. That means that in the absence of a binding partner, at least there is no single structure for that protein. So an example of that is this protein I showed you the model of before. This is the NMR structure of it, so part of it is beautifully folded, but the two ends, they have no structure isolated in solution. Maybe they would get some local structure interacting with something else, but just the protein alone, these are intrinsically disordered regions, and they have no single structure to be modeled. So AlphaFold will give you a structure, it will give you the coordinates back, but in a sense, it's not a failure here that these are low confidence, it's simply a reflection that there is no single correct structure to be found for this intrinsically disordered region. And people have looked at this a little bit more. You can look at this distribution of these PLDDT scores, and you can see that the vast majority of low PLDDT scores are actually disordered regions. We can predict those by different methods, and we can see mainly down here, where you get a low quality prediction, it's because it's intrinsically disordered. Going a step further, it turns out that looking at AlphaFold models and these low quality prediction regions and inferring that they actually represent disorder, as in this case, turns out to be a very good way to predict intrinsic disorder. It's quite an elaborate way. There are much simpler sequence methods, but it's certainly as good as the best of those methods for predicting intrinsic disorder. That's just one example, actually, of AlphaFold being used for a purpose that's by no means what it was originally designed for. So moving on to the third output from AlphaFold, you get the coordinates, you get the local quality, you also get the PAE, or predicted aligned error. And to illustrate what this means, I've chosen this example from DeepMind's own website. So if we look at the bottom protein first, we get the impression it has one single large folded domain here. It's got this sort of floppy, irregular loop structure in the middle. The PAE is a two-dimensional plot, which says that if I superimposed the target true structure and the model on residue X, how confident am I that residue Y is where that superposition said it should be? So I do the superposition on residue 100, residue 200 in the model, and the true structure are 10 angstroms apart. How confident am I that they are genuinely 10 angstroms apart in the true structure? So you read these as the relative confidence that regions of the model are placed correctly relatively to each other within the structure. So if we look at this one, the dark green here says that there should be a low error in that predicted alignment, in the distances between residues X and Y for residues that are part of this big folded domain. So it's in two parts separated by this loop. The dark green says that for residues 80 to 340, we're very confident that they're all totally reliable with respect to one another. The same for this big block here. And then this block here, when you think about it, says that I'm confident that this first block here, represented by 100 to 300, is correctly positioned with respect to the second block from 400 to 800. And that's exactly what you see here. So you would read this as being there's one single confident globular domain here. It's interrupted by a long flexible loop of low quality where we don't know what's going on. That's what these stripes are here. And there's little bits at the termini that are less clear as well. But we basically have one globular domain. You compare that with this figure at the top, what we've got here are kind of four distinct regions. So from 0 to 80, we're very confident that's one block. Everything is well positioned, confidently positioned within that range of residues. Likewise for this block here, and this block here and here. But you'll notice off the diagonal, we have no confidence at all that this first domain is placed correctly with respect to the second domain. That would be this region here, with respect to the third domain, or with respect to the fourth domain. That makes total sense when you look at the model, because each of those four domains is connected by a long flexible link. So in solution, all of those domains would be totally independent in motion with respect to each other. And indeed, AlphaFold has correctly said, I may be confident about the individual structures of the domains, but don't ask me to be confident about how they pack together. I really haven't a clue. And that's correct in this case. So if you think about it, you've got the coordinates, you've got the PLDDT that tells you about the local quality, and you've got the PAE, which is more about the global properties and whether individual regions are packed correctly with respect to other regions far apart in the sequence. Okay, so that's what AlphaFold is, what it produces. How can you get AlphaFold structures? Well, I'm going to give you three ways. The first is the easiest. That's looking at a database that the EBI has produced called the AlphaFold Protein Structure Database. It's grown from its first incarnation, which covered just proteomes of important species like human, pathogens, E. coli. until very recently, just over the summer, it now covers 200 million proteins, which is 90% of whole of UniProt. So for most of UniProt, there is a model in the AlphaFold database waiting for you. Curiously, viruses are an exception, and I believe that's because viruses are often expressed as polyproteins chopped into individual chunks, and the polyprotein is what's encoded by a UniProt code. So viruses are not there, but more or less everything else is. Interestingly, just to note in passing that the emergence of this, the reporting of this database was not without some controversy. So both Nature and Science sort of poked the ant's nest in the choice of wording. So initially, Nature said AlphaFold has determined the structures, and that's, of course, that's not the case. These are merely predictions. So they rapidly produced a correction. In Science, they said AlphaFold has revealed the structures, and that's not really true either, because they are still predictions. Some of them will be right, some of them will be wrong. But nevertheless, this is an amazing resource, provided you remember they are all predictions. So you can actually go to the database and plug in a UniProt code, or you can plug in some text and try and find models of your protein that way. You can also do a sequence search. That's very easy to do. The EBI has a suite of different ways of searching based on sequence, and they can all plug into the AlphaFold database as sequences available in the AlphaFold database. So it's very easy to find models. If you want to search with a structure and find similar structures to ones, something you're interested in, then it's a bit more complicated. DALI is the best method really for this. It's the best established. People understand all of the statistics, but it is a bit slow. And so what they've done on the server is allow you to search the AlphaFold database, but only one species at a time. And that might be very useful if you're interested in human. You can search all of the human proteins in the AlphaFold database with the DALI server. One method that came out quite quickly to try and be quicker and allow you to address the whole AlphaFold database was this, based on a quite clever idea of representing the shapes of proteins in an efficient way. But this turns out to be fast, but not necessarily very sensitive. The one I'd really recommend, which has come on the scene and made a big splash quite recently, is called FoldSeq. So this combines essentially speed and sensitivity. So it's almost as good as DALI in performance, but it's pretty fast. It's still not quite fast enough to scan the whole of the AlphaFold database, but it scans a redundancy reduced version of it. Just to give you a flavor of the sort of the clever methodology that allows them to search the whole database, or a large portion of it, very quickly. It works by comparing strings. So in a way, that's analogous to what you would do with a database search based on protein sequence. But the strings are actually representing local structural neighborhoods rather than amino acids. And so the way this works is that for each residue in a protein that you're encoding in a string, you look for its nearest neighbor in the structure, you measure some kind of geometrical properties. These have been defined and sort of squashed down to 20 different states. This is just a coincidence. These don't correspond to amino acids, even though it's 20. There's 20 different structural environments. And so each of the residues in this protein would be represented by the nearest structural state of the 20. And then, of course, once it's a string, you can use very fast algorithms to compare a string that represents a protein that you're interested in with a string that's been pre-calculated for a model in the AlphaFold database. So it's very quick. It's also just recently introduced a very, very useful feature which allows you to filter what you're searching for on phylogenetic terms. So you can put in a species, or you can put in a group like reptiles, and it will only search within that group. And that will be even quicker and give you a more selective list of results. So I think FoldSeq is, well, it is already, and it's going to be a major structural bioinformatics resource. This is what it looks like. There's a server, it's very easy, free to use. You decide on your database. There are 2 ways of scoring. That's not so important. This is that really useful taxonomic filter. So if you want to search just in mammals, you can. The results that you get look quite a bit like you might get on a blast page. You get these representations of the models that match yours. You get a list, you get statistics, you get the alignment, you get a structural superposition that you can just twiddle around. So just moving on, keep an eye on the time. So the database is great, but you'll see in a second, there are many times when you want to calculate your own models, and you can do this very easily, largely thanks to the work of this guy, Sergei Ovchinnikov, and his group, who have started, set up these resources called AlphaFold. These are ways in which you can essentially run AlphaFold in slightly variant ways I don't have time to go into in your browser. You paste in the sequence, you have parameters you can choose to change or not, and well, half an hour later, perhaps you get back a model. The reason this works is because it piggybacks on Google's cloud resources. They have a system called Colab for collaborative programming, and this piggybacks on that to produce models for you. So we sometimes talk about impact in academia. The impact of setting this up and making AlphaFold available through a browser can hardly be overestimated. It's huge. So Colab obviously gives you the models. It gives you them colored in this now familiar way that tells you that the PLDDT. This is the PAE that you'll recognize. And we'll talk about these other ones. I see I'm slightly short of time. It is totally free, but there's a little bit of potluck in which cloud GPU you get. So sometimes you can run out of time, your job can fail. In that case, you can pay for extra, or you can consider running it on your own machine, which is what we do. It doesn't have to be a super fancy machine, but it does need a GPU and plenty of memory. So for around £4,000, you can get a machine that will make quite large models in a few hours. Limitations. So AlphaFold is great, but when you come to use it and apply it to different proteins, you realize that sometimes it doesn't give you exactly what you want. So you may have to work a bit harder. So we all know that proteins have different conformations. Those conformations can be important parts of the way they work. Here's a transporter in 2 very different conformations. If you send that sequence to AlphaFold, you may get back this structure, or you may get back this structure. Maybe you get back the one you want, maybe you don't. And that there's a little bit of potluck. You may need to work harder to try and get alternative conformations, and I'll describe that a bit later on. There are dramatic proteins that have fold swapping, which is essentially 2 even more divergent structures. And again, AlphaFold is going to struggle. It will produce one of these perhaps, but maybe not the one that you want. Multi-domain proteins are hard. You may not get the conformation that resembles your crystal structure for a multi-domain protein because the domains may be connected by linkers, as in this protein that's, I think, 12 zinc fingers linked together. So AlphaFold is always going to give you, it will always produce a structure, but we have to remember, of course, that for these proteins, there are many different possible structures. So there isn't a single correct structure for AlphaFold to find. AlphaFold doesn't know anything about membranes. So here's a transmembrane model, and it's partly fine, but it's also got this funny little mini helix and tail of protein that's in the membrane, or where the membrane would be. And it obviously shouldn't be. The AlphaFold doesn't know that. It also doesn't know about post-translational modifications or ligands. We'll come on to that. I mentioned at the beginning that the covariance from the multiple sequence alignment is important. What that means in practice is if you've got a sequence orphan, where you've got a protein that doesn't have any or many homologs in the database, then you can struggle to get a good model. but at least it will tell you that the model is not very good. And also, you may know something about your protein. You know, you may have been in the lab and seen that it's got a disulfide between two cysteines. You can't actually feed that information into AlphaFold and ask it to take into account other information you have. There are also some limitations of the database in particular. Right now, so surprisingly, it has a few very weird errors that have this odd technical explanation. I think I'm just mentioning this because they're there right now, but they should disappear within a few weeks. Of course, there's only one, I say of course, but you won't know that necessarily. There's only one isoform, so many proteins have multiple isoforms. What's in the AlphaFold database will be a model of the longest one. You may be interested in a different one, so therefore you'll need to make your own model. AlphaFold database, where proteins are very, very large, small number of huge proteins, it splits them up, so you may not find them easily. They are available with a bit more difficulty. Of course, there's no oligomers or complexes. There's only single chains in the AlphaFold database. So if your protein acts in a complex or as an oligomer, you need to look elsewhere to try and predict that structure. And then finally, what I've put as better models may be possible with today's sequence databases. What I mean by that is that the structures deposited in the database were calculated on a particular date, at which time there were a certain number of sequences in the protein sequence databases. And if you're unlucky and you have a target where there's only a small number of homologs available, then the database will contain possibly a poor model, as here. That doesn't mean that in a year or two's time, you won't be able to make a very beautiful model of this protein, because by then perhaps the sequence databases have expanded and you can build yourself a bigger, more diverse alignment. So it's not clear yet how often entries in the AlphaFold database will be updated. So you may want to make a fresh version in case the one in the database is a bit old, essentially. So I see I'm almost at the question time. If we're still on track for questions, I've taken slightly longer than I expected, but here's a comparison just to summarize some of the differences between getting your AlphaFold structures from the database, getting it through the web page, and getting it locally. And there are pluses and minuses of each here. So I think I've mentioned most of these negatives of the database. It doesn't have complexes. So complexes you can build with AlphaFold, I'll show you in a bit. But you need to obviously do that yourself, either on the web page or locally. Obviously, the database has a single confirmation, so you can, if you need to sample, try to sample alternative confirmations. You need to do the modeling yourself. I think I've talked about the others. Yeah. So at that point, I will see if there's any questions, see if the chair thinks there's time for questions or if I've rambled on far too much already.
Yes, we have actually a couple of questions coming in already. I'm not going to take all because I just want you to finish on time. So I'm just only take two questions. So the first question is, you know, is the quality of the AlphaFold prediction is related to the length of the proteins, the size of protein, or not?
No, not so much. No, it's one of the remarkable things. You know, the previous generation ab initio modeling methods had a very strict size limit. Above 200, you were really struggling. These, you can plug in thousands of residues and they will produce good models. So the answer is no, really. Simple as that.
Okay, great. The second question before you move to the next session is, are the outputs, especially the PAE, similar for AlphaFold multimer or not? Or what are the drawbacks of this software compared to the monomeric AlphaFold predictions?
Well, I mean, the multimer, I'll come on to it. It's a retrained version of AlphaFold specifically for building complexes. It's a good question. The PAE that you get from complexes, I'm not sure I can answer that right now. Actually, it's I'm not sure. I think you probably get to PAE for a single chain. I'm not sure it's clever enough to recognize the confidence of relationships across the complex. Although I'll show in a second, there are other protocols that give you scores to try and help you decide whether an interface is actually reliably predicted or not.
Okay, great. So Dan, you can go ahead, and we actually have lots of questions, so we can go through all when you finish.
Yeah, okay, that's fine. Sorry, and I'll try and accelerate. I always do this. Right. So moving on to the uses of AlphaFold, what you can and can't use it for. Structural biology is a big deal. So most crystal structures are solved by molecular replacement, which requires a prediction essentially of at least part of your unknown structure, not unknown, your remaining to be determined structure. Then you try and place that prediction within the asymmetric unit, derive some initial phases, and work your way up. So people are full of stories about old data sets that they've only managed to solve with the benefit of AlphaFold models because they tried and couldn't solve it any other way. So it's been a revolution in the area of molecular replacement. This slide just notes that quite often in more complex cases, you will need to split a multi-domain prediction up into the individual domains in order to place those domains accurately. So this just introduces in passing RosettaFold, which is a kind of competitor to AlphaFold, or it was inspired by AlphaFold. It came out slightly before AlphaFold. Overall, not quite as good, but worth trying alongside AlphaFold, definitely. And you can see a multi-domain protein. The domains are very beautiful, but the orientation is not quite right. So you need to split them up, and then they fit perfectly on the target, and you can solve your structure. And you can do the same for fitting into even quite low resolution cryo-EM maps as they did here. So there are some tools, actually. I just mentioned these in passing. These are things that we've developed ourselves. These are part of CCP4. If you're using AlphaFold 2 models or looking in the database with a view to something to solve your molecular replacement, there are some useful tools we've developed. MRParse is over here. This looks in the AlphaFold database at the bottom panel, also the PDB as it happens. So here AlphaFold database hits for your protein. And I've just shown this example because remember I said there are very few errors in the AlphaFold database. This is one of those cases where lots of the similar proteins, mainly the modeling has worked fine, but there's one real dud for that technical reason I mentioned before. So you can see that straight away from the color coding and obviously avoid that. We've also got this software slice and dice to address complex multi-domain structures to split them up. That splitting them up either aids molecular replacement or cryo-EM fitting. So you can see here that the overall structure captures the helical repeat quite nicely, but it's not quite superimposing well. We can trim off all of that red spaghetti automatically and split using a variety of algorithms the prediction into chunks. Those chunks then fit very well and solve the structure quite easily by molecular replacement in a way that the original model doesn't. So these are tools to help link between AlphaFold and structural biology. Also, just to mention, this is from another group, the Phoenix group. They're also software developers. They have this interesting idea of an iterative approach where you take your AlphaFold model to your map, you do some work with it in the context of that map, manual or automatic, and then you take that improved version back to AlphaFold, provide it as a template, ask AlphaFold to have another think using that as a basis, and then you can take that back to your map again. And so you can kind of alternate between the map-aware kind of refinement and AlphaFold for perhaps working with filling in gaps that are not obvious in the map, working with difficult regions. And you can, there's a Colab page where you can do that, linked to the Phoenix suite for structural biology. I won't go into details here because it would take too much time, but I mentioned before that AlphaFold database contains one conformation. Quite often when you run it yourself, you'll get 5 models, but they'll all be the very similar conformation. If you want to sample conformations more broadly, then there are a number of tricks that you can use. So this might be the case, you've got a transporter in a membrane and you've got the outward facing structure come out quite easily from AlphaFold, but you want to get the inward facing structure. What can you do? Well, you can introduce a random element, a stochastic element into the AlphaFold calculation using these flags here. So this is an advanced version of the Colab page, and it has instructions that show you how to trigger an element of randomness and produce more models within the calculation. And so by sampling more broadly, you may get out a greater variety of conformations. Another option, these are in order of ease, this is the easiest, this is the hardest. Another option is to, sounds very odd, the MSA, the alignment that gives you the covariance is important, but people have realized that if you deliberately reduce the depth of the MSA, you can cause AlphaFold to produce different models, essentially. You can also push AlphaFold in the right direction. Remember, it takes templates. So if you've got a template in the right conformation and AlphaFold keeps insisting on producing the wrong conformation, you can provide AlphaFold with that template in the right conformation and try and get it to pay attention to that template. And that might mean ignoring other features as well. And then I don't think I'll even mention this one. It's quite hard to do, I think, quite manual at this stage. So if we move on now to say, okay, I've got an AlphaFold model, I want to use that to interpret or to predict the function of my protein. What can we say about the quality of the AlphaFold model for that purpose? Can I use that model in the same way as I would use an experimental structure? And if you think back, we've got that quote from the organizers of the CAST that said in some cases AlphaFold structure predictions were indistinguishable from experimental ones. The key point here is in some cases, right? So yes, sometimes the models are good enough to use, but you need to be confident yourself in that. You need to have the confidence to interpret those models and be sure in yourself that you're working with something that's reliable, either overall or particularly in the area that you're interested in. And just to mention some kind of benchmarking, some kind of equivalence that people have drawn between the quality of AlphaFold models and other kinds of models. So here they basically said that AlphaFold is as good as an experimental NMR structure, so a rule of thumb. Whereas here they said that where you've got a crystal structure at 3.5 angstroms or better, then that you should use that instead of AlphaFold structure. So these give you some kind of benchmarking about how AlphaFold models compare against experimental models of different kinds. If you're working with a transmembrane protein, it's worth mentioning that you can do some extra validation using this database here. What this does is say, okay, this is where I think the membrane is for this AlphaFold model. How does the content of the bilayer compare with what I predict to be there from sequence methods? You know, does it have extra bits and pieces that shouldn't be there, or is it missing a helix that was predicted to be transmembrane but is not sitting in the bilayer? And they've got these 10 further criteria to help you assess whether the transmembrane, alpha transmembrane protein model is good or not. So how can AlphaFold models help you with function annotation? I mean, if you're confident in the quality, how can you use them? Sometimes the models simply produce a surprise domain. So this is an example of a protein that we took an interest in. This is quite clearly a transporter of some kind, but this domain was a big surprise. It wasn't obvious in any kind of sequence search. Turns out to be a gold domain, which we can then link to the way that this protein transfers from one part of the cell to another. Here we've got a model, looks like a very good model, very dark blue, that's beautiful. We think it's probably a phosphatase that acts on this. So you can treat that. If you're confident in the model, you observe this dark blue, you can more or less treat it as you would a crystal structure or an NMR structure, and you can go about assigning function, inferring function. So here is where the pocket is, that's the binding site, and sure enough it's conserved and it's positively charged because the substrate is negatively charged. So essentially, if you've got a very high quality model that you're confident in, you can treat it with a whole range of methods that are available for working with experimental structures. While we're talking about ligands, there's a kind of follow-on database from AlphaFold called AlphaFill, where they've kind of tried to add in missing ligands. So here's a globin. We know that all of these globins bind heme in roughly the same way. Here's the model. We can recognize that the model has a homologue in the PDB. We can see that we can compare the structure of the local binding site for the heme, recognize that it matches quite nicely what's in the model, and therefore we basically transfer this ligand into the AlphaFold model, and that's what you can find in the AlphaFill database here. This is another database trying to do more or less the same thing. And I'll move on quickly to talk about this one. This is another area that people are very excited about. It is in silico screening for drug leads. So if we have the structure of a drug target, can we use it to pick out compounds from a big database that might be inhibitors, for example? That's work that's pre-printed here. So what they did, what was a key part here was the editing. So in this case, it's colored brown, but this is very low quality stuff. And in fact, there was a low quality region lying right across the binding site for this protein. So this is where you need to be careful, right? You would need to say, okay, I can recognize that this part of my model is unreliable, it's interfering with my analysis, I will remove it. That will be essential for docking your ligands into this pocket here, because otherwise this low quality region is in the way. Once you've treated them carefully, then they're actually as good as apo structures, so ligand-free structures, in terms of enriching, in terms of searching through a big database of compounds and picking out things that bind to the protein. These AlphaFold models are actually as good as the crystal structures, but not as good as the crystal structures that have ligands, because the ligands induce some local changes to the pocket of interest. So you can use them for these purposes, but with care, and that's key. One question that's been particularly controversial actually is whether we can use AlphaFold models to predict the consequences of single nucleotide polymorphisms, these variants. And there was quite a high profile paper came out that said, no, you can't. And to be fair, the reason that they said no, you can't, is because that when you make a model of a mutation like this, which should be very damaging, because this arginine is not going to be happy in the same place as the proline, what you get back is initially quite disappointing. You see the structures look exactly the same, and you think, well, that can't be right, because I know this should be very destabilizing. But it's not quite as simple as that, because if you have a high quality structure, as we do here, dark blue, high PLDDT, trustworthy structure, we can use that to provide the structural context that allows us to do better bioinformatics predictions of the stability change. So the red here means that indeed that arginine mutation has been recognized as destabilizing, with the benefit of the structural context provided by a confident AlphaFold 2 model here. And there's this bioarchive paper, so saying exactly that actually, that if the model is good enough, you can use it as you would a crystal structure and predict in a structure-based fashion what the consequences are of a mutation for the stability of a protein. So you can use it to help understand SNPs. And actually, there's an interesting final detail as well, which is that even though the structure looks exactly the same, The PLDDTs are quite different in this region. The proline looks very happy. The arginine, even though it's in the same place, these local PLDDT scores are visibly different. So it's as though AlphaFold is sensing that there's a problem here. Even though it doesn't know where to put the arginine any different, it knows that it's not happy there, and it's reflecting that in these PLDDT scores. There was a question about complexes. AlphaFold and related methods can co-fold complexes. This, to me, is one of the most amazing things that came out of AlphaFold. Within a few months, RosettaFold came along, and by simply throwing 3 sequences at RosettaFold, it can predict not just how each of those individual proteins folds up, but how the 3 of them come together to form a trimer. I mean, absolutely amazing stuff. And people very quickly made AlphaFold do the same. So these are high quality predictions out of AlphaFold, better than docking individual predictions with the best docking methods. And this is AlphaFold Multimer that the questioner mentioned. So AlphaFold proved capable of co-folding, producing these predictions, but that wasn't what it was designed for. So quite quickly, DeepMind went away and retrained AlphaFold to produce AlphaFold Multimer, which is also available through CoLabFold, and that does a better job of predicting these complexes. And this is just also relates to the question. There's a number of different protocols that people have developed on top of AlphaFold. The reason I mentioned this one in particular is that it gives you this score, which helps you understand whether the model that's been produced, the interface within that model, is actually reliable or not. So you don't get this score directly out of AlphaFold, but if you run AlphaFold in the context of this AF2 complex, you can get a score that's very helpful in telling you reliability of the interfaces. And you may have seen this, people have used AlphaFold for very, very large complexes, modeling a lot of subcomplexes, fitting them into quite low resolution cryo-EM maps. Absolutely amazing work, and an AlphaFold integral to that. This is an interesting little nugget of something that AlphaFold can't do. AlphaFold is not aware of symmetry, and people have commented on this from the beginning. One of AlphaFold's rivals, Unifold, has been developed with a symmetrical version. What the advantage here is that even in very complex structures like this, internally, it's working with what is called the asymmetric unit here, and the symmetry is factored in without the complication and the memory requirement of modeling something as big as this. So this is just out. Again, you can see how important the CoLab initiative was. Here's another CoLab for Unifold. And this was interesting, just in passing. You might think, oh, how many proteins are actually symmetrical? Well, if you look across the PDB, almost half of them have some kind of symmetry. So actually the answer is an awful lot. And I know I'm running out of time, but it works with protein-peptide interactions as well. Interesting thing to note there is that there are 2 strategies you can use, actually. You can use the protein and the peptide separate, or you can use the protein and then a long polyalanine linker and then the peptide sequence. And if you're really focusing in depth, you should try both approaches, because there's the idea that they are actually complementary, that sometimes one will work and the other one won't. And then, while we're talking about things AlphaFold can't do, it can't yet do anything with nucleic acids, but this absolutely amazing work from the RosettaFold people, just in the last couple of weeks, steps into that area, allows you to predict nucleic acid structures or even protein nucleic acid complexes just by providing the sequences. I mean, it's mind-boggling. It really is. You might be looking at this and saying, well, that's not actually a very good prediction, but RNA structure prediction is particularly hard, and this is actually well ahead of the state of the art. Not yet available as a server as far as I know. So to conclude, AlphaFold is obviously a massive step forward, but I've been saying throughout, you need to have the confidence to be able to interpret the results, to edit those models, to remove bits that are going to complicate downstream analyses. And you also need to be able, ideally, to know of ways to go beyond the simple AlphaFold. So if it doesn't come out with the conformation you want, or you want to build an oligomer, then you need to work a bit hard. And so you need to be familiar with options for going beyond AlphaFold. Of course, AlphaFold will accelerate because of all of the relevance to molecular replacement, to cryo-EM fitting, but it's not going to replace experimental structure biology. And as you've seen, I hope there's a whole ecosystem of these methods going way beyond what AlphaFold was designed for. And actually, AlphaFold has inspired other people to produce competitors that plug some of the gaps that AlphaFold still can't address. And last slide, I promise, just to kind of, where are things going next? Well, there are other methods for ab initio modeling which appear to be as good as AlphaFold in terms of the results, but maybe much quicker because they work in different ways. Surely things like post-translational modifications will come to AlphaFold at some point. And I don't have time to talk about it, but protein design has been absolutely catalyzed by the deep learning approach of ab initio modeling. You can use the similar kinds of network to start with a random sequence and iteratively produce a completely novel protein. Amazing possibilities. So I'll stop there. Apologies for running slightly over. I'm happy to take any questions if people are still around. Can't see anyone, of course.
All right. Okay, you can see me. Yeah, we actually have lots of questions. So yeah, if you have time, everybody stay. We have a lot of interesting questions coming out. And we're also gonna finish later with some summarized presentation as well. So I think one of the, I just apologize ahead that we may not be cover all the questions you ask, and your question, if it's not answered, will be emailed to Dan, and we can have a further discussions. And I think just pick a few, I think it's quite important question is, what do you think of using AlphaFold structure in MD stimulations? Is it sensible to try transmembrane protein stimulation if AlphaFold is unaware of the membrane during the predictions?
That's a very good question. I mean, AlphaFold's not aware during the prediction, but of course, you as an expert can look at that prediction and say, well, that part shouldn't be in the membrane. I will sort of move it out, or I will go to an alternative structure prediction program and try and get a different result, or I will try and produce a different conformation by one of those tricks as I saw. So there's nothing, I would say, transmembrane proteins bring their own problems, but if you can address those problems by being familiar enough with those issues and sort of solving those problems, then yes, I don't see why you can't take the result and do molecular dynamics on it. Of course, you would want to be confident that the structure was high quality before you did that. I think to take a medium quality structure and hope that molecular dynamics would refine it into something more beautiful is very, very unlikely. But if your structure is largely blue, high quality, then that whole rationale of why not treat it more or less like an experimental structure, then yes, that applies, I would say, to dynamics in the same way. But like I say, take those special care with the transmembrane issue.
Okay, great. There's also a question about the chemical environment. So do you think AlphaFold actually take into account of the chemical environment around the protein, for example, a low pH region to predict protein structures?
No, AlphaFold doesn't know anything about its environment. Short answer. I mean, if you were looking at pH behavior, then I think it would be molecular dynamics, but that comes with all of the provisos that's starting from a reliable starting point.
Yeah. So is that sort of also related? Does it also take into metal binding? Is it good for metal binding prediction too?
Yeah. Well, yes. I showed you that example where binding sites are well predicted, even though the metal is not present in any way during the folding.
Yeah.
And what that means is that you can interpret, if you suspect your protein binds a metal, you can go and look in your model and see where, there are some cysteines that might bind a zinc. But there are also methods, including deep learning methods, coming along specifically to take a structure and try and predict where a metal binding site might sit. So absolutely, try these things.
Great. There's another question which I think is maybe rely with lots of people sort of wondering is there any need to refine the AlphaFold structure? Like if they predict it, do you actually need to look at the geometry, whether they are good to go?
No, not really. The geometry will be, well, apart from the low quality red spaghetti, that's quite funny in a Ramachandran plot kind of backbone geometry sense, but the folded regions, they will be geometrically favored by definition. It's funny, our role last time in CAS was exactly to say, to look at refinement methods. And it was just as other people were saying, wow, these AlphaFold models are amazing. One aspect of that was we came along, we said we can't refine them anymore, or rather, when we look at the efforts of refinement groups to improve on the AlphaFold model, they can't improve it anymore. So it was basically, it had hit the ceiling. So no, there's no need to refine or attempt. There's no real point in attempting to refine an AlphaFold model.
Okay, great. Follow up with lots of questions, but I think maybe just take one or two more. One of the things is sort of, I think a lot of people as a structural biologist interested, what's our role as experimental structural biologist? So is there any sort of, you think, is going to be useful to prioritize the experimental project protein that structure is weakly predicted by AlphaFold and to increase the knowledge of the structural space?
Yeah, that's an interesting point. I mean, I think it does. I think the availability of these predictions will make structural biologists perhaps redirect their work a little bit. I mean, a few years ago there was the whole structural genomics exercise where people tried to plug gaps in fold space, and if there's an unknown family out there which produces a very reliable looking model with AlphaFold, then in terms of what does it look like broad fold, I think that's kind of job done. But of course, that doesn't mean that if that's a drug target that you don't want them to have the added value of a crystal structure. So I think it will, I think it depends what you want the structure for. I mean, if you need the fine details, then there's no substitute for a high resolution crystal structure. If you just want to fold up a protein because it's a representative of an interesting new family and you get a reliable model out of that, then that's probably your answer, really, in many cases. Yeah.
Okay, so I think it's just so many. We're gonna contact you through all the questions. I just wanted to finish in this seminar. I don't want to drag everybody too long. So I would like to thank you, the speaker, Dan, for the wonderful talk, and everybody who asked a question which has not been answered, someone have been answered. And we can continue the conversation through look at the Twitter account for Biochemical Society and Portland Press. And we welcome suggestion for any future topic. If you're interested to be featured in the webinar, and you can send a proposal through the websites. And you can also find a lot of recording of previous webinar, and also current one will be online on the website and YouTube channel as well. So you can find lots of resources. And I wanted to draw your attention that Biochemical Society 2024 awards are now open for nominations. It features a new portfolio of streamlined nomination process. It has never been a better time to nominate outstanding peer or colleague who deserve the recognition. And the deadline for initial nomination is 1st of November 2022, with the submission welcomed by both member and non-members. So please visit the website to find out more. So finally, I would like to highlight that in this very challenging times, it's more important than ever to stay connected and to engage with each other. And it's exciting time to join the Biochemical Society with all the community researchers, specialists together and stay connected and take advantage of the key benefits, including discounted registration fee for society courses, meetings, and exclusive access to the wide range of the grants, students' bursary, personal online access to 2 of the journal, and more. And for me personally and my group, it's benefit from this membership significantly. So that's it. I just want to thank you again to everyone, and hopefully to see you in our next seminar. Thank you.